coming. My name is Maggie Schultz. I'm the education coordinator here at the Kickapoo Valley Reserve. So thank you guys for being here for our next Driftless Dialogue. I just have to give a quick shout out to the Ralph Newsom Lecture Series who helps fund these talks. Thanks to a grant from them, the Kickapoo Reforestation Fund through the UW-Madison College of Agriculture and Life Sciences, and the Friends of the Kickapoo Valley Reserve for helping make these happen and that we can provide these to you for free. Today we're really excited to welcome Cristina Dahl from the US Fish and Wildlife Service. She works at the Midwest Fisheries Center in Onalaska. Just a little background on her. She's the outreach specialist there. So she graduated from Virginia Tech with a bachelor's degree in wildlife conservation. She interned for the US Fish and Wildlife Service throughout college, and after college she worked as a park ranger at the Necedah Wildlife Refuge. So as the outreach specialist for the US Fish and Wildlife Service at the Midwest Fisheries Center, that's quite a mouthful, she gets to share a lot of cool information about fish. So hopefully you guys are here to hear about some fish. So thank you for being here.
Yes, thank you. Thank you. Yeah, so as you heard, my background's in wildlife biology. So when I came to work at this fisheries center, I was really amazed at all the work that you can do with fish and all the technology and the science that they do at my center. So hopefully that's exciting to you guys too. I just wanted to start with introducing my agency. So a lot of people have never heard of the US Fish and Wildlife Service. Has anyone here heard of the Fish and Wildlife Service? Okay, I'm impressed because I hadn't heard of it until I interned. So you're one step ahead of me. But our mission is working with others to protect, conserve, and enhance fish, wildlife, plants, and their habitats for the continuing benefit of the American people. We are also the agency that regulates the Endangered Species Act. So if there's an animal that may oh, that was weird. Sorry. That might be considered to be endangered, we're that agency. The federal government is confusing, so I made this hierarchy, but basically our agency is housed under the US Department of Interior, and here are all our sister agencies. A lot of times people get us confused, but as you can see, we have like the national parks, the Bureau of Land Management, USGS, which is also in the La Crosse area. And then within the US Fish and Wildlife Service, we do a lot of different things, and I could probably sit here trying to explain all of that. So we do things like communicating what we do with the public, which is really important since we're funded by our taxpayers. The ecological services is more of seeing if species need to be threatened or endangered. Office of Law Enforcement, we have staff at airports who are making sure people aren't poaching wildlife. And then our National Wildlife Refuges are kind of our public lands. And then there's also the Fish and Aquatic Conservation branch, which I'm a part of. So a lot of times we get confused with the DNR, and we do a lot of similar things, but our scope is much broader. So we work across the US, so we have offices and public lands in every single state and US territory. So if you go to Puerto Rico or Guam, you can find a National Wildlife Refuge there too. And then more locally, I often get this question of, do you work at the Necedah National Fish Hatchery or the refuge? And those are also part of our agency, but they're just meeting our mission in a different way. So that's our public lands. And then the fish hatchery is actually also part of the Fish and Aquatic branch, but they do more of like raising fish to release, whereas we are a fish technology center. So we are one of 7 fish technology centers in the US. And basically what we do is we do science and research in hopes that it can be used out in the field. So under our Midwest Fisheries Center, and I know there are a lot of hierarchies, but that's the best way to explain it. Oop, whoops. I don't know why it keeps doing that. We have 3 main offices, which is our Fish and Wildlife Conservation Office, our Whitney Genetics Lab, Fish Health Center, and then we also have a data branch, which is new. Basically, we created that to modernize our agency, modernizing the federal government. It's a task. And so pretty much what we're doing there is sometimes we have people who have worked for our agency for decades and they are the expert on this species and they have so much institutional knowledge, but then they retire. No one knows where their files are. Nobody can read their data sheets. And so that's just us trying to solve that solution with the data branch. And then we also have an outreach program, which is what I do. So if you're interested in environmental education programs, we do that for free and you can come talk to me. But for this lecture, we're going to focus mainly on the work of what these 3 offices do. So our Fish and Wildlife Conservation Office, again, is split into 2. And so we have a native species and habitat branch and then an aquatic invasive species branch. And either branch, what we're doing is we're catching fish. So this is something that I thought was really interesting is all the ways that you can catch fish. So one way is you can use a pike net. And that basically funnels fish in. They hit this long piece, and then here's that hole that's really tiny and makes it easy for them to get caught, but really hard to come back out. Sometimes we also use electrofishing. So these string things actually have an electrical current, and those go into the water and they temporarily shock the fish, and then we have biologists scoop them out. It's really fun. Probably one of my favorite things, the gill nets are pretty much exactly like what they sound. Ooh, sorry. Thank you. So basically you set that net across a long area and any fish that's swimming through that area will hit this net and get caught in the gills. And then we also have something called seine nets, where basically you just kind of scoop up anything in that area as well. And that one is a little bit more physical, I guess, for the biologists. And so there are a lot of different cool ways to catch fish. Probably the coolest thing is you never know what you're going to catch when you put things in the water. So we put nets in the water and caught just dragonfly nymphs just full of them. So that's always been really exciting, or when you catch a really big fish like a paddlefish in the Mississippi. But every time we are catching fish, we are always collecting data on species and size. Anyone have a guess on why those 2?
Age?
Yeah, age is definitely a factor. Yep.
You want to know the species so you can keep track of distribution?
Yeah, that's exactly when we're the agency regulating the Threatened and Endangered Species Act. That's definitely something. So yeah, you guys kind of hit on some of the points. So indicator species, those would be species like brook trout. If you caught a brook trout, you could probably assume that the water quality is really high. Brook trout are really, really sensitive species. And so if you have a breeding population, then you have to have high quality water. Invasive species, and I'm sorry, my slides are a little off here, but we are also the agency that is monitoring a lot of invasive species, and with a lot of invasive species, the earlier you find them, the better the results will be, rather than when they just kind of have reproduced and it's really hard to control that population. And then threatened and endangered species, just like Maggie mentioned, if we don't have any data on fish, how are we going to know that 20 years ago their population was super high and now all of a sudden it's plummeted? And then age class as well. So a lot of times we can tell if an area is overfished. If you're not finding any big fish, that's going to tell you that it's overfished. If you're not finding any young of the year, that could also tell you something, that maybe there's not enough food or maybe there's a disease. So yeah, that's all. We're a science-based agency, so we work off of our data. Within the habitat branch, they do the work with endangered and threatened species in the area. And anytime we're working with endangered or threatened species, we're asking the question, well, why are they endangered or threatened? Here you can see a lake sturgeon, which is not federally endangered or threatened. It is under review to be listed as an endangered species, but that decision hasn't come out yet. I think it's a great example. Oops, here. So this species is a great species to think about life history. So basically understanding that this fish can live up to 150 years. So think about that. That's enough for 6 generations of your own family to potentially encounter the same fish. It is also huge, so it can grow up to 8 feet long. And one of the most important things that we needed to understand about this species was that it is a migratory species. So they have very specific spawning requirements. So they need a certain water temperature, they need the flow to be a certain way, and then they also need the stream bed to have big boulders in them, basically so that they can lay their eggs and the eggs just kind of fall in between those boulders, and then they don't have to protect those eggs because they're just naturally protected. But because of those requirements, they often have to migrate long lengths to find those habitat requirements. And so we found that dams are actually one of the biggest impediments to their populations and actually causing declines in their populations because they're not able to successfully spawn. Like a lot of threatened and endangered species, there's also that historical aspect of things. And so this fish was seen as a trash fish because it would get caught in people's nets. And so they would just catch them and pile them up like logs and then burn them. And then eventually people realized that their eggs made really good caviar, and then they got overfished because of that. Okay, so here you're gonna see how we tag fish. So this is actually at the Necedah National Fish Hatchery, and they insert something into a little tag into its nostrum, and then you were seeing a little metal scanner just to make sure that the tag's in there. So we work a lot with the Necedah National Fish Hatchery to stock up lake sturgeon in areas where they previously were. This is just one way that we can keep track and see if that stocking is successful. So if you caught a fish and it has that little tag, then you know it was from the Necedah National Fish Hatchery, and we can measure their progress over time. We also have different tags. So we also have different types of tags. So the tag you saw in that other video doesn't tell you much other than if you caught that fish and you see it has a tag, you know it was from our hatchery. But we also have other tags that are actually able to identify an individual. And then that way you can record its growth and how it's doing over time, which is pretty cool because lake sturgeon are such long-lived species, and they also spawn pretty late in life, that's really important to understand them over a long time. So some of our biologists actually works to modify and remove dams. This is really important for migratory fish, but a lot of dams in the US have been decommissioned or need a lot of maintenance, and it's going to take a lot of time to fix them. Some of them don't even work for their purpose anymore. And then they can also be a huge safety hazard. So they have a recirculating current that causes a lot of people to drown if they don't know. And so she works with communities all across the region to modify and remove dams. And so this is one of those scenarios where she modified them. Maybe they still need that structure, but now fish can actually migrate through there. So win-win. Another thing, or another animal that we work a lot with are freshwater mussels. They are an incredible species. I do a lot of work with students and kids about freshwater mussels, and I always show them this model and I ask, is this an animal? Because when you see this, I don't know, would you expect a complicated animal to live in this shell? I love opening this up. So it's a great model of how complex freshwater mussels are even in the inside. So they even have hearts. This would be its heart here. Interestingly enough, its digestive tract also goes through its heart. Really interesting. Whoops, I'm doing this wrong. It's the pressure. Okay, whatever. And they also have stomachs. Here is the gill, and then this thing right here is a single foot. Freshwater mussels are really, really important for our waters, and I have a little video here to illustrate why. What I was going to show you, so this is a screenshot of the after the video, but these two tanks are filled with water that both look like this. And here, let me actually, they both look like this, but this tank has freshwater mussels in them. And so after a couple hours, this water turns into this. So they are incredible filters of water. They can filter out E. coli, they can filter out pharmaceuticals, hygiene products. Filtering out the sediment is really important for fish specifically because they're breathing out through their gills. So when there's a lot of sediment in the water, that's causing them stress. And if they're stressed, that can sometimes open them up to different diseases. So having clear water is really important for us, for wildlife, for fish. And it's actually a known thing that whenever you have a healthy community of freshwater mussels, you also have a healthy community of fish. Another interesting thing is when they filter anything that they can't eat, they actually spit out into like this little mucus thing, and then there's some food for the fish. So we saw that mussels have only one little foot. How do you think they move? How do you think they spread all over the place? They're all over the Mississippi. Does anyone have any guesses?
Do they attach to something else?
They do, yes. So their life history is probably the coolest thing about freshwater mussels. So different species have developed a part of their body, which is called the mantle, to look like something that a fish would eat. So some freshwater mussels look like fish. You can even see the eye. You can see how they have a spotted pattern to kind of look like, I don't know, maybe a trout. This kind of looks like a snail to me. And then some look like worms. This is really grainy, but that's supposed to look like a crayfish. And then I think this also looks like a snail. So they're trying to attract a fish to actually bite onto them, because once the fish bites onto them, they have their young, which are called glaucidia, which are tiny little Pac-Man freshwater mussels, and those actually attach onto the fish. It can attach to their gills, or it can attach to any part of the fish's body, and then they'll grow and develop. So here's a nice close-up picture, and then they drop off. And so that's how they are distributed all over the rivers. And then let me show you this really cool video. Of how that works. Very cool. I've had students ask before, they asked, well, how does it know what to turn into if it doesn't have eyes? How does it know to look like a fish? I don't know the answer. So since learning about freshwater mussels, they're definitely one of my favorite animals now. Unfortunately, they are one of the most endangered animals in the whole world. And so some of the reasons they got that way is the historical context. So especially on the Mississippi, they're really, really beautiful on the inside, and I can pass these around. But someone thought these would make good buttons. And it was said that you could find freshwater mussel beds from stream bank to stream bank of the Mississippi. And even just look at that, that is so many mussels. And I just can't help but think, what would the Mississippi look like if we still had that many mussels? It's crazy. Another reason we work on dams is because they also affect freshwater mussels. So each species has a different fish host. So maybe one species needs a bass, one species needs a walleye, and so on. But when you have dams, you're potentially separating freshwater mussels from their fish host, and then they're not able to reproduce. Another reason why they're endangered is pollution. Specifically, they're very sensitive to salt. So if you're using salt on the road, you probably hear this all the time, but it's just another reason to make sure you're being conservative and still being safe, but they're very sensitive. They're filters, but they're still affected by things like nitrogen and phosphorus. Like a lot of things like wetlands, they filter out, but at a certain point, it gets too dirty that they can't even really function. And then zebra mussels. So zebra mussels don't have that really cool, unique life cycle where they need a fish host. And so they reproduce very rapidly, and sometimes they can suffocate freshwater mussels. So habitat is really important. Things like channelizing rivers, sometimes that can be so, or the current can be so fast that all that sand can bury a population of freshwater mussels. And with a lot of endangered species, a lot of them are endangered due to habitat loss. But at our office, we have a biologist, Heidi Cooler, who works with Fishers and Farmers Partnership, and she works with farmers and landowners who want to change farming practices to benefit streams and water. It's really cool. So basically they give technical support like surveys on birds and macroinvertebrates, and also fund these farmer-led groups, which is also really cool. It's not a top-down approach. It's having the farmers work together and see how they can influence a watershed. And so they do things to like cover crops and prairie strips, and they have been really successful. So I think I was reading today in some of these groups, they've reduced the amount of nitrogen and phosphorus in the water by 90% through prairie strips, and reduced soil loss, topsoil loss also by 90% just through these practices. So that's a really cool part of what we're doing, is that we're also focusing on that habitat part. So now let's talk about aquatic invasive species. Anyone know what this fish is? Yes, yeah. So our agency, we actually call them invasive carp. But when we say invasive carp, we're talking about these 4 species. So silver carp are the ones that jump. Grass carp disrupt vegetation. Big head carp, they're usually the first to colonize an area. And then black carp actually eat things like mussels. So we're really concerned about that in the Mississippi River. They were introduced from Asia. They are filter feeders. And so someone put them in their pond thinking that I was going to get rid of algae, but then of course they got released. And so now they're a big problem. As far as things that you can do to just make sure that we're not spreading them more, is sometimes they can be found in live bait. And it's really hard to identify them because fish so small look so similar. So a really important thing that you could do is throw away your bait. Don't release it in the waters, just an easy thing. One cool thing in terms of aquatic invasive species are the technologies that we use to understand these species. So one of the things that we do is split beam hydroacoustics, and basically what that's doing is it's taking a picture of the water column, and you can see how big schools of fish are, and you can even see how big individual fish are, which is really important for understanding how successfully an invasive species is invading the area. Another thing is, in the same way that wildlife biologists use collars to track and monitor animals on land, we actually use acoustic telemetry, which is what this is here. So these things basically talk to each other. This is called a receiver, and then this is a transmitter. We can't use GPS underwater, so the way that this works is each one of these transmitters has a unique sound, and anytime it passes by this receiver, that receiver picks up the sound and can tell what individual tag is what we call it. What is crazy is here you can kind of see the evolution of technology. So this is what we used to put in fish. This would hang out. Here's another one. Now we've kind of, well, here's another in between. So that's a little smaller, still hanging out though. And then we got down to something like this. And now we're actually down to something like this, but all of those tags are in use. So I can't bring it. I only bring them when they're broken. So to do that, we actually have to perform a small surgery on fish. Who knew people did surgery on fish? I had no idea. And so they just do a small incision and insert that. That has been really important for invasive carp in the Mississippi. One of the problems that we have, though, is people don't know about this technology. And so they'll just find this weird thing that looks like this in the river and think, what is that? And I guess they don't, maybe they'll read that, but they don't know what telemetry equipment is. And so a lot of people will sometimes destroy these receivers. And we have a lot of receivers all over the Mississippi River. Because as the fish swim, that's the thing they're communicating to. Sometimes they also look like this. So this is a real-time receiver, And this one actually uses satellites and cellular data so that anytime a fish swims by, that data is actually going to the biologist's phone in real time, which is why it's called a real-time receiver. Recently, in December, we started using this technique, which is called the trader fish. So basically, the fish that has this tag in it is the trader fish, meaning in the winter, they are a little bit slower and they tend to, why am I blanking on the word? They tend to be in groups. That's really basic. Yeah, school up, thank you. And so that's the ideal time to catch them. But how do you know where they are in the big Mississippi, the third largest river in the world? And so that's when this technology really comes in handy, because if one fish is schooling with the others, it can tell everyone else where we should put our efforts to catch these fish. And that actually happened near Trempealeau. We caught 300 carp, and that was the largest catch ever recorded in Trempealeau. So we keep using this technology, and if you happen to see it, let your friends know, because it's helping us keep our river healthy. Another thing that we do, which the Whitney Genetics Lab primarily works on, is eDNA, which stands for environmental DNA. So what that means is basically fish in the water column are constantly shedding scales and slime and secreting feces. And so all of that has their DNA. So if you take a water sample, you can actually catch that, and you can test and see if their DNA is present. It's a lot more complicated than I'm making it sound. The actual sampling for the DNA is as simple as scooping up some water in a test tube that looks like this. What is complicated is that eDNA, the technologies that we use can be so sensitive, so there are a lot of protocols about if you touched a carp, well then don't wear those clothes, and we have to have separate equipment for carp because then that could cause a positive detection. And then you think about all the lab stuff that goes into that, but I'm not going to bore you today. But basically, after spinning it down, this is your little bit of DNA here. We not only use this for invasive species, but we also use these for endangered species like the Topeka shiner. They're a great species to use eDNA for because they like oxbows. Everyone familiar with oxbows? So basically, rivers meander and they keep getting wider and wider, and eventually they get so wide that they kind of cut off and the river decides to take the fastest way and it keeps going straight, but there's basically almost like a C shape left in the landscape. And there can actually be incredible fish communities in those oxbows, and Topeka shiner actually prefer them and need them for wintering habitat and spawning habitat. Those oxbows are kept alive by the flooding cycles, and eventually if it floods so big or so much, it brings more animals or they're able to get out. But basically, because they're confined to such a small area, the chances that you would get a false positive of eDNA are very slim because it's such a small oxbow area. They're so rare that the chances that an eagle ate them and dropped it nearby in that oxbow are pretty low. And you're also not having the issue of maybe there's eDNA upstream and it's flowing downstream. So they're an awesome species for eDNA. It really helps us. So if you think of an area and you have a bunch of oxbows, we're always thinking about staff time and resources. And so if you take the time to do eDNA, and you have areas that are positive, then you're probably going to focus your areas or your work on those oxbows. Another really fascinating thing that we're getting a lot of funding to do is actually called metabarcoding. So in eDNA, this is kind of what happens. If you have a sample, it probably has a lot of DNA, but you're just looking for one species, and so you amplify it through this process, and you make a lot of copies of the DNA, and then you're able to tell if that species is in that sample. But metabarcoding, instead of focusing on one species, your water sample can tell you every single species that is in the water. So that can be really helpful for invasive species. So let's say there's an invasive species in Michigan, but not yet in Wisconsin. This could tell you that maybe they're in these waters. And it also has implications for endangered and threatened species. So you might be able to find new populations that you weren't able to find before. So it's really cool. The future is now. And then our last office is our Fish Health Center. And they work a lot with fish hatcheries, which are prone to disease because anytime you have big or large numbers of animals in a confined space, you have to worry about disease. And so what they do is they go around to our national fish hatcheries and tribal fish hatcheries and test them for diseases. That's really important because if you're releasing these fish into some water, you want to make sure they're healthy and you're not getting the wild fish sick. Another thing that they do are wild fish health surveys. So there was, now I'm not going to remember the name, but there was a disease that was killing a lot of fish up north, and it kind of came out of nowhere. So the idea is they just go out and catch wild fish and test them for different pathogens just to know what's out there. And that database is actually available to the public if you're interested. So a lot of times when they're sampling, they just take a sample out of the kidney because the kidney's filtering all those pathogens, and so it's going to be really concentrated. And then here they're actually testing for bacteria, and so this medium has sugars in it, so if there is a bacteria, the bacteria will eat it and grow, and then we'll do further testing. Another really cool thing that we do is diagnostics. So if there's a novel disease in fish, or maybe there are a bunch of fish dying and nobody knows why, these are the experts. They're incredible. They even create tests for novel diseases. So like, you know how COVID came and we didn't have a test and someone had to develop it? That's basically what they do for fish. We also have biologists who study parasites, fish parasites, which are really important. So they can be really good indicators of biodiversity. Lots of parasites need a lot of different hosts. So this is a parasite called black spot. So if you've ever caught a fish and it has black spots on it, that's a parasite. Most parasites, as long as you cook your fish, you're going to be fine. So you should cook your fish. But this one, as you can see, uses a bunch of different hosts. And so that's why we're studying parasites. Because if you have a lot of different parasites, you probably have a lot of different hosts that it's using. And so you probably have some great biodiversity. And with that, I know that was a lot, but if you have any further questions, I think we have time for questions. Yes, yeah, I saw your hand first, yeah.
When this eDNA sampling, it indicates the presence of a species, but can it tell you also the relative concentration of the species? In other words, comparing, we have a lot of this and a little bit of that?
No, it's mostly used as a presence-absence tool. I would say if you happen to get a lot of positives in an area, that would tell you that you most likely do have a positive. there is an issue of like maybe you have a positive, but it's a false positive, and so you have to do further investigation. But from my understanding, it can't tell you that. Yeah. Yeah.
The Chicago, that carp thing, what's the status?
Yeah, so right now they're really trying to prevent carp from invading the Great Lakes and the river in Chicago. Basically, that river is really, the Illinois River is very invaded with carp. And so they have a technology, it's almost, I think it's like a sound gate. And so the sound deters the carp from moving into that area. And so as of right now, there is not a breeding population of carp in the Great Lakes. Yeah. Yeah.
We live on a little creek, comes out of springs and then it goes in. And in the springtime, about now, we get this foam on there. And I get conflicting answers.
Okay.
It borders on, oh, it's nothing, it's natural.
Okay.
But my suspicion is there's something going on. Have you seen that foam?
I think I know what you're talking about. I know sometimes I've heard that foam can be plants breaking down. But yeah, I'd have to see it. I don't know if you have a picture or
Our creek is about
anything.
that wide.
Okay,
And
yeah,
It'll be foam from here to that window.
yeah,
Then the nearby town, it bubbles over the top of the dam,
huh.
and then the river from here to the wall is.
Yeah, I'm not sure. But yeah, I mean, I wonder, maybe you could find a resource to find some free water quality testing, and that could tell you, like, if you're concerned about nitrogen or phosphorus, there's a great program. It's called the Water
Which
Action.
one of those two foam?
I don't know, honestly. I'm sorry, I wish I knew. But I know that program is free, and if you signed up, you might get the materials to do some water testing if you're curious. But is your water, it's near a spring?
Is it what?
It's near, like the spring is feeding
Our water
that?
supply? No,
Oh,
not-
okay.
Not near that. Creek that has that.
Okay.
It's the creek that goes
Yeah.
Okay,
down.
yeah,
It ends up in the West Baraboo.
okay, yeah. Sorry, I don't have a better answer for you.
It'll be about that. It'll be that.
Yeah. The foam is that thick? Huh. Yeah, that's very interesting. Yeah.
Disturbing to me.
Yeah, that's very interesting. I've never seen that except for the ocean, but yeah. Oh, sorry.
Why did they pick Onalaska, the place to have the fish hatchery?
What's that? Can you repeat
Why
that?
did they pick Onalaska, Wisconsin, they have that fish hatchery? Why wasn't there the Mississippi? Why that area? So there's some in the country, right? So why did they pick that? How long has that been there? And do you give tours?
The Necedah National Fish Hatchery?
No, yours.
Oh, mine?
Why did they pick that area?
That's a great question.
Well, I don't know how long it's been there. And
Yeah.
you give tours.
So I believe we've been there. We used to be near the Riverside Park. So there, it's actually now a wedding venue called the Hatchery. So that's where we were a long time ago. But then we moved to this area, and I can't even tell you when, but it's been a while. And so the refuge used to be housed out of there, and ecological services. We are open for tours. So if you're interested in seeing our labs, is the main thing. It is an office building, so I wouldn't have the expectation of seeing nature. Yeah. Well, not anymore. We did have a wet lab where we can do, well, specifically our fish health center. They will do disease challenges on live fish to see, I don't know, testing. Maybe they'll introduce a disease to a live fish and see if a treatment works for them. Or we're also testing drugs for fish. So like at a fish hatchery, if they get sick, we have a veterinarian who prescribes different medicines. And so then our office also tests these medicines to see if they can be used. So sometimes we have live fish, but I don't think so right now, if you come. Yeah. Yes, yeah.
Do you test fish for like contamination of mercury or like in Madison, they warn people to only eat fish twice a week out of Monona Bay or something?
Yeah.
Do that kind of testing?
We don't. We mainly test for viruses and bacteria. Sometimes in our wild fish health surveys, I don't think we're doing parasitology tests, but it's mainly bacteria and viruses. Yeah.
You probably don't test for microplastics then either.
We don't. No, we did have a grad student who worked with us, and he was researching microplastics in freshwater fish, but not specifically at our office.
Yes.
Yeah. Yes, yet. Yeah.
I have another question.
Yes.
How much does one of those receivers cost? The ones that people destroy?
So one of these things, whoops, I think is $200. And then one of these things, I would say $250 plus, probably more.
Are the receivers anchored down somehow, or?
They are.
Their boats aren't getting them, or people don't catch them with their, well, do they catch them? When they're fishing?
Yeah, I think mostly it's out of curiosity they pick them up. So I'll go back to my picture. But our biologists actually created the special anchor for these because it's such a niche thing that it wasn't created. So it looks like this. And so then the receiver gets put into this metal thing and secured that way. Yeah, so, and then usually this will be tied to a tree or something. So it probably does look very intriguing. You're like, huh, what's that? And you pull it up. But that's one of the most important things is that these actually have to be completely underwater to work. And so that's another way that people can kind of disrupt that work if they just pick it up and they're like, huh, weird, and just throw it on shore. So yeah, we're really trying to spread the word that these are really important in helping the Mississippi River.
Is your
Yeah.
name on them or something? Is it marked?
Yeah, so we do have this tag. That's another hard thing because even if you had a piece of paper, that's not going to survive in the river. So we have this little thing, but I guess it's not convincing. Yeah. Yes, yeah, that's a good point. Yeah. Maybe we should put a little note like conservation work. So yeah, we are working on how we can make that a little bit more obvious. We did have an idea to maybe have a sign on that tree of like, what am I? But then sometimes I'm like, well, would that bring more attention and more problems? Probably.